So, very good evening. Today we discuss about the some life histories, structure, and life histories of some Pteridophyta members. Here, in the xylotom, we discuss Lycopodium, Selaginella, Inquisitum. So, these three, four species, we discuss about the structure and life cycle of the these Pteridophyta members. Okay. So already we know that one here xylotom is belongs to the Pteridophyta member, that is the Xylopsida. Xylophyta or Xyloptera division is there. The class is Xylotopsida. Order is the Xylotails. The family is the Xylotaceae family. The genus is the Xylotom. Okay. So these pteridophytes possess an independent, well-developed sporophyte stage. We know that one. So the sporophyte here, the diploid condition is there, which is the sporophyte is the independent plant, and well-developed vascular system is present. In this plants exhibit a great diversity in structure and reproduction system. So in this gametophytic stage is very quick, independent and dissimilar from the sporophyte stage. So most of the ferns and the terrestrial in nature, a few species are in aquatic, or some are the xerophytic condition is there. So the alternation of generation is conspicuously, and heterologous condition is present. So so here the classification of the xylotom. So please tell to join the class. Your friends also is no name. I don't know about that one. Please join the class. So some members are joining, and remaining also please join. Okay. Mm.
Good evening, sir.
Very good evening. So tell to your friends to join the class. So put a message. Okay. Is audible? Is clear?
Yes, sir.
Okay. uh, okay. So here the morphology of the xylotom here in this xylotom here two species are there, that is the xylotom nudum and xylotom flaccidium. These two species are there. Now we see the external feature of the these xylotom here, the plant body, which is In rootless plant, we know that one. The rootless plant and brown rhizomes are present. So, which is the rhizomes are there, and the aerial shoot system is also present. Means the not well developed root system is present. That rhizome is there, horizontally rhizome is present. On that horizontally rhizome, the aerial branching system is present. So around here 20 centimeters height there. That aerial branch system means the shoot system is green in color, and the rhizome is slender dichotomously branched, which is covered by the hair-like brown rhizoids are present. Rhizomes, they are bear the rhizoids. So these rhizomes and rhizoid to underground position and horizontally they are to develop. So these rhizome with help of the rhizoids, they are to fix in the soil. And from that rhizome, they are erased the shoot system, which is in green color around 20 centimeters in height. So some fungal elements also grows in these rhizoids, which is help in the nutrition. Okay, and some of the dichotomous branches of the rhizomes are grows out the soil, which is developed into the erect aerial green shoots. So already you see here. Uh, so this is some green shoot system is present. Okay, so there are the both rhizome and aerial stems are continuous and are morphologically is identical. So that is the rhizomes are in brown color is there, colorless or sometimes brown in color, and the shoot system is the green in color. So the aerial shoot means nothing but stem is the multangular and ribbed in the lower part. See here on the picture, the diagram here, see that picture. In the multangular structure is there. Okay, in that here xylotum nodum, the terminal parts are the quadrus, while the flattened in the flaccidium. So there are two species are there, xylotum nodum and xylotum flaccidum. So these two species are there. There are the morphologically the dissimilar these two species. The stem is photosynthetic stem and small scale like appendages are there. So these small like appendages we call as a leaves, and with are the vascular traces and irregularly arranged in the stem. So in the terminal plot, the leaves are replaced by the fertile and the sterile appendages are there. On the apical region, some are the fertile and sterile organs are there. These fertile ones bear the synangia. See here, that yellow color, the triangular structure is there. That triangular structure we call as a synangia, which is the reproductive organ. These produce the spores. Okay, so synangia is the reproductive organ, and which are the called sporophylls. These sporophylls is differ from the vegetative leaves. So those leaves the bear the sporangia. That leaves we call as a sporophyll leaves. And some leaves are not produced the sporangia. That leaves only doing the photosynthetic part only. Okay, so the sporophylls are there. The fertile appendages receive vascular bundle up to the base of the sporangia, while the non-vascular connecting to the leaves and sterile appendages. Now coming to the, is clear? Is audible?
Sir.
Okay. Right. Thank you. So Coming to the anatomical structure of the xylotum. So here, mm, so which is the differentiated to rhizome, aerial shoot, and sporangia. These are there. So see here, this picture, the lower part of the that in brown color is the rhizoids are there. That is the rhizomes and rhizoids are there, which is goes horizontally. On that rhizome, they produce the erect shoot system is there, that is green in color, dichotomously branched. Upon that, small appendages also there. That appendages we call as a sporangia. So these sporangia, which bear these sporangia, that leaves we call as a chlorophyll leaves. Okay. So coming to the internal structure of the rhizome, here there are the a large rhizome possesses well smaller about the in 1 millimeter diameter is present. So which is the differentiated into cortex and steel. So cortex and steel is present. So when we take the transfer section of the rhizome, here two parts we can easily to identify that one. The outer one is cortex and inner central region is the steel is present. The cortex is differentiated into three zones are there, three parts are there. That is the outer thin-walled parenchyma is there, which is the have endophytic mycorrhiza is there. Endophytic mycorrhiza means these fungi is association with these xylotome to fix or to observe the phosphorus element. So these xylotome give the shelter up for the fungi. So both are the symbiotically association with the mycorrhiza means mycorrhiza and these xylotome. And the outer one is these parenchyma cell, and middle one is also parenchyma cells, which is the store the starch storing region or storage region, and the inner cortex, which is the dark brown pigments are there. So this one is have the tannins also present, and condensation is there. Condensation means oxidation, and condensation is there means very dark in color is there. So here, so that's why we easily to differentiate into these three regions in the cortex. That is the outer cortex, middle cortex, and inner cortex. So here the endodermis with the typical Casparius strips also present, which is easily to demarkable to the cortex from the steel. So inner layer we call as a, that is the endodermis is present. And the steel is surrounded by a single layer of the pericycle is also there, which is followed the phloem. The phloem consists of elongated parenchyma cells that are the is made up of the lignified content is there, just like higher plants. So lignified tissues are there. Okay. And the phloem is surrounded by the central xylem, which is various sizes according to their diameter of the rhizome. Depending on the rhizome size, the xylem is present in that size. Okay. So here the tracheids possess calariform thickness and it is absent. So that's why we call as the steel is the protosteel condition is present. Okay. Protosteel condition is present in these stylotum rhizome. So when we take the aerial shoot of the xylotum, we see that anatomical character of the shoot. When we take the cross section here, three different regions are there. That is the epidermis, cortex, and centrally steel is present. Okay, is clear? Is audible?
Sir.
Okay. So here, huh? The shoot system we take the in the shoot system we take that one. Here, three regions are there. Outer one is the epidermis, which is the protecting layer is there. Middle cortex and centrally is the stele is present, which is the vascular system. So here the epidermis is well developed and heavily cutinized condition is there. Epidermis is the outermost layer, which is the single layer and which is covered by the cuticle is present. Well developed cuticle is present, so that's why which is to stop the transpiration. Okay, so means they have possess some xerophytic conditions also. Well developed cutinized is present. They are possess the xerophytic character is there. That one. And the sunken stomata is also present between the longitudinal ribs. Sunken stomata is also one character of the xerophytic connector meant to stop the transpiration. So coming to the cortex here, the below the epidermis here cortex is present. So this cortex is the parenchyma tissue, which is the differentiated into three zones are like that rhizome. Here outer one, middle one, and inner cortex is there. So outer cortex, which is around 3 to 5 rows cells are there, which is made up of the parenchyma. Means chlorenchyma is there. Means pigments are there. Chlorophyll pigments are there. So that's why the shoot system, which is appear in the green in color. So due to the chlorenchyma, number of means plenty of the pigments are there. Chlorophyll pigments are there in these cells. So, and the outer zone, the middle zone here around below the outer zone, middle zone is present, which is around 4 to 5 rows of the cells are there, which is made up of this sclerenchyma tissue. So, due to this sclerenchyma, here they give the mechanical strength to the that shoot. Sclerenchyma is present, means narrow lumina is there. So narrow lumina and there is no intercellular space and thick cell walls are there. So lignified chlorenchyma means sclerenchyma is there. Okay, so that's why they give the mechanical support to the plant. And another one is the inner zone, which is consist in thin walled parenchyma is there. That is they have the intercellular space also, which is the storage region. Means the star deposits are there, and the innermost cortex, that is the endodermis, is conspicuously, and they have the Casparian chips also. So Casparian chips also present in these endodermis. And steel is, we can see the protosteel condition. Protosteel means without pith. Already yesterday we discussed that one. Protosteel means the xylem and phloem is continuously they are arranged without pith is there. The centrally parenchyma, the pith is absent in this protosteel. And here the pericycle is not well developed. The phloem consists of a well vascular elongated cells are there. The xylem is surrounded by the phloem in star shaped. So star shaped, that is the xylem is there. Okay, xylem is mm, that is the star shaped xylem is well. So centrally is some metaxylem is also present, and some sclerin fibers are there in that centrally. Okay, so that's why here the siphonostylic condition is also present. The leaves are here very minute in structure and lacking the stomata and vascular supply. So, which is a single layer epidermis is covered, which is the cuticle is also covered. That is the uh, anatomical character of the rhizome and shoot system and leaf also. Is okay clear?
Yes, sir.
This is okay.
Yes, sir.
See here, so this is the stellar system. Star-shaped steel-like structure is there. See here. Huh? Uh, this one. Okay. So coming to the reproductive structure of the these xylotum here. So this is the synangium. Synangium, a pear-shaped synangium is there. This is in yellow in color. That one. The plant body of the xylotum is a sporophyte, which is fertile and sterile region are not demarcated. So the axial of the shoot system is small appendages, which is the globular structures are there, which is called as a synangium, and which have the vascular bundles for the food supply. So there are the there are the sporophylls are there. There are the short lateral branches, which is bear these sporangia, these synangium, and these are the uh, in the synangium is externally trilobed condition is there. See here, when we take the section of these synangium here, which is look like a trilobular. See here, sorry, trilobular condition is clearly we can see that one. See here. So this is when we take the transverse section of this synangium. Here, three lobes are present. Trilobe condition is there, and there are the so these phalanges are triad condition is present. There are to fuse to form a single synangium. So based on the long peduncle, they are consist as a sporangiophore. So developmental stages suggest which are the locule of this synangium arises from the single epidermis cells are there. See here a single epidermis cell is present to protecting layer. So these sporangias fuse form to a synangium. So here this synangium, which is the jacket layer, is there. Below that separating jacket layer, a sterile tissue. That sterile tissue, they have the archisporial tissue. So these archisporial are sporogenous tissue to develop as a spore mother cells, and these sporoderm cells to develop as a meiotic division to produce the spore tetrads. Okay, so all the spores are similarly alike, means all spores are similar and smooth in condition. Sometimes they have the a fine reticulate ornamentation is present. The epidermal layer of the jacket became thick walled except the small vertical slit which are to use for the dehiscence of these spores. When they complete the development of these spores, huh, so here this areas is slit these sporangia. These that one we call as the stomata or uh, opening area of the synangia. Okay, clear?
Yes,
So
sir.
then the develop is okay, clear?
Yes, sir.
Yes, sir.
Thank you. So So, so after the hey, okay, cool. So when they complete the development of these spores, so there are the liberated and to produce as a prothallus. Means prothallus is the initial stage of the gametophyte. Is the class we discussed that one. So here the haploid means after the meiotic, the spores are developed, which is the haploid condition is present, and which is small brown gametophyte will develop. The prothallus is cylindrical, around 1 to centimeters in long and 1 to 2 millimeters in diameter. They have the branches dichotomously in nature, and the each branch terminate into a single apical cell. They are to cut into four phases. There are the pyramidal apical cells. There are the three cutting phases are there. See here, the structure of the gametophyte here triangular triad condition is present. So already we discussed that one. When we see that one, the see here the triad nature is present. See here. So the prothallus is pale yellow to brown color, and they have numerous hair-like rhizoids which are appear on the surface of the prothallus, and rhizoids also develop gradually. And these prothallus the differentiate into uh, number of division to develop as a gametophyte. So the prothallus consists an outer small cells which are the star deposits, but the some lacking the mycorrhizal fungi in this one. And in the coming to the reproductive organ here, when we take the gametophyte here, the xylotom or monoecious conditions, which are the antheridia and archegonias are intermixed and they cover with the entire surface of the prothallus. Okay. The antheridia here, see here, mm, the antheridia. So antheridia, this one is the so the antheridium here, the mature antheridium is in spherical and projecting out from the surface of the gametophyte. The jacket is formed with a single layer of the cells, which is consist some androcysts, which gives rise to the around about 200 of the anthrozoids. These anthrozoids are multiciliated and spirally coiled, which can be released by the breaking upon the opening cells are there. When coming to the archegonium, these archegonium here flask-like structure is there, which is consist of the venter and neck canal cells are there. The neck they have around four ventral rows, which are 4 to 5 cells height surrounding the one or two neck canal cells are there. The venter canal contain the egg and the venter canal cells. So this is the solid based condition is present. And when the development of the egg, the ventral canal cells are degenerated to passage is to form the entry of the anthrozoids. So after the fertilization means where these anthrozoids release from the antheridia, they are swim to reach the egg to form a zygote, and the zygote divide transversely forming the an outer epibasal cell toward the neck side and inner hypobasal cell. So the epibasal cell again divide number of divisions to give the rhizome, which is the aerial roots are produced. So hypobasal cell gives us the food as the axis develops from the epibasal cell, shoot apex is towards the next side. So here this one is uh, the development of the embryo. Okay. So the life cycle, when coming to the here, the life cycle, there are the alternation of generation is present, means the xyloton sporophyte, which is the produce the sporangia then produce the spore mother cells. The spore mother cells undergoes to the meiotic division. This is to produce the spore. So meiospores are spores. So the spores to germinate in the feasible environment, settle in the favorable condition, they give the prothallus. So upon the prothallus, they are to bear the antheridia and archegonia. There's antheridia is archegonia. There are produced the respective gametes, and they are to fuse to form the zygote. After zygote to develop as the embryo, that embryo again to develop as a young sporophyte condition. So this is the alternation of generation. Means only the gamete is very less condition is there, less life cycle is there in their xylotum. Means xylotum is the sporophytic plant body. Already we discussed that one. There is the diploid sporophytic condition is there. So that's why there the spongia is to produce the sporophyte only. Okay, is clear? Okay, are you clear? The xylotom.
Yes, sir.
Thank you. So coming to the next type study of the in Pteridophyta, which is the lycopodium. So here the lycopodium, see here, this one is the lycopodium plant. Means lycopodium is commonly called as the club mosses. Huh? Club-like structures are there, so that's why we call it the club mosses. So coming to the classification of these lycopodium, we can see the division is the Lycophyta or Lycophyta. The class is Eligulopsida. Order is the Lycopodiales. Family is Lycopodiaceae. Genus is the Lycopodium. So the member of the Lycopodium, around 400 species are there in the tropical and subtropical region of the worldwide. They are commonly called as the club mosses or ground pines. Commonly called as the club mosses. Why we call the club mosses? Here, the plants of the apical pod, the club-like structures are there. That is the cones. See here, this one, stobalus or yellow color stobalus are present. See here, are you clear? Yellow color, a thick cells are there, thick leaves, sporophyll leaves. That sporophyll they are aggregated to form a cone-like structure. Chobalas or club-like structures are there, so that's why we call it the club mosses. And ground pine also, pine-like structure is present, so that's why the name is coming. Their structure club mosses or ground pine. So these mosses, they are look like a pine-like structures are there. So coming to the morphology of these lycopodium, here this one is the creeping, some are erect and scrambling species are there. There are differentiated into two species are there. That is Eurostachy and Rhopalostachy species are there, based on their morphological feature of the lycopodium. Is it clear? Is audible my voice? Okay.
Yes.
Okay. So here Urostachy and Ruphalostachy, two groups are there based on their morphological appearance. So coming to the Urostachy species, the plants are erect in terrestrial forms and pendant in the epiphyte. So coming to the Ruphalostachy, so these are the creepers. Creepers are there. Ruphalostachy plants are creeping like structures are there. Urostachy plants are the erect stem is present. And coming to the Urostachy, the roots only at the base of the stems are present. But Ruphalostachy roots are adventitious type and lower portion are present. And bulbs are formed for the vegetative reproduction in the that is the Urostachy species. These bulbs are not present in the Ruphalostachy species. Okay, so here only the sporophylls are involved in the reproduction system. So here the branching is dichotomously branched in the Ruphalostachy, and in Urostachy here dichotomously branching is there, but the creeping it is appear as a monopodial branching is appear. Monopodial like structure is appear this one, and the root system here very advanced and advantageous root system is present in this lycopodium. Okay, so here that leaves around in the microphyllus lanceolate condition is there, which is broad sessile basis. Okay, microphyllus and lanceolate shape. The leaves are the lanceolate structure is there, and the broad base is there. There are the sessile leaves. Sessile means there is no petiole. Okay, there is no petiole. They are directly attached to the plant in stem. So that's why the petiole is absent. And so they are arranged spirally. They are spirally on the stem. On the stems are there. There are four rows around the four rows in the stem. So the fertile leaves bear the sporangia, so they called as sporophyll leaves. So those leaves are arranged these stabales are sporophylls. So that leaves we call as a sporophyll leaf, and they bear the sporangia. So, so here the leaf-like sporophylls bear the kidney-shaped sporangias are there. These sporophylls are smaller than vegetative leaves, especially in the advanced species. The sporophylls are aggregated into cone-like structures are there. Chabalas. Okay. So they are called as a chabalas or cone-like appearance is present. Uh, so this is to form a cone-like structures are there. So that's why these leaves are aggregated to form a chabalas. So that's why they called as a chabalas. Uh, okay. So these leaves are these branches are dichotomously branched. So coming to the internal structure of the this lycopodium here. So coming to the internal structure of the lycopodium. So from the roots, when we take the transverse section of the root, here there are the three zones are there, that is epidermis, cortex, and centrally stele is present. So the outer epidermis, they have the some root hairs are there, which is the protecting layer. Below the outer epidermis, a cortex is present. This one is a parenchyma tissue. Sometimes sclerenchyma is also there. And centrally, the stele is present. That stele is monarch, diarch condition is also there. Some species which is appeared in the U-shaped diarch is there. Okay. So when we take the transverse section of the leaf here, there are the outermost epidermis is there with number of stomata are there in the upper or lower surfaces. The stomata, okay. So here, this is the dimorphic condition, and also there some Lycopodium verbally. So the parenchyma cells are angular without intercellular space. So here midrib is the central, that is the endarch xylem is present. When coming to the transverse section of the stem, so stem is very important in your exam point of view. Transverse section of the stem of the Lycopodium, they are asking in a essay question, which is compulsory asking. So when we take the transverse section of the stem, here there are the distinct regions are there. There are the differentiated into three regions are there. That is the epidermis, cortex, and stele. Epidermis, cortex, and stele. The epidermis, cortex, and stele is present. The stele is present. The epidermis is a singular epidermis. They have some stomata also present. See here, the stomata is also there, and that is the cortex is well developed. Cortex is there, which is the parenchyma cells. So cortex is divided into three zones are there. So that is the outer cortex, middle cortex, and inner cortex. Okay, so that is the outer cortex. They have the sclerenchyma tissue, which is give the mechanical strength of the that plants. Outer and inner cortex. These two cortex they made up of the sclerenchyma tissue. So in the middle cortex, they have the um, parenchyma tissue, which is the oval shaped or circular shaped, globular shapes are there, which is the give the storage region. Only the outer cortex and inner cortex they give the mechanical support. And the below epidermis means innermost layer of the cortex. Here endodermis is present. A prominent endodermis is there. So below the endodermis, stele is present. The stele is here. Outer pericycle is also there. Below the pericycle, the centrally see here, the xylem is present. The xylem is surrounded by the phloem. So here there is no pith. So that's why the stele is here. Protostele is okay. Clear?
Yes,
Okay.
sir.
So the stele condition here, protostele condition is present. So here, the stele here there are the diversified steel is present. So based on the arrangements of the xylem and phloem, structure of the xylem and on the xylem and phloem arrangements, here there are the different types of the steels are present. So actinosteel based on the actinosteel is there, and actinosteel means here the xylem is there are to formed as active means star-like shaped is present. So that's why here we call as actinosteel. Okay. So here the xylem is to form a star shaped around 5 rays. 5 rays is differentiation is there between these 5 rays. The phloem is arranged between these rays. So here, so that's why that steel we call as a actinosteel. And another one is the polyarc. So another one is the polyarc condition. Polyarc actinosteel. Polyarc means more irregular arrangements are there. Okay, irregular arrangements and number of xylem rays are there. And another one is the plectosteel. Plectosteel. So plectosteel means patches like structures are there. Bar like structure means xylem is broken into plates. Number of plates are there. The xylem is breaks into number of plates are there. Between that parallel bar, the phloem is present. So that one we call as a plectosteel. Okay, Nandi. So this plectosteel is to arrange in the that is Lycopodium serratum. Lycopodium serratum, we can see in this plectosteel. And another one is the mixed protosteel. Mixed protosteel we can see in the Lycopodium serratum. So means here each species they have the a particular type of the steel. Okay, depending on the species here the particular type of the steel is arranged. Okay, so mixed haplosteel or mixed protosteel is there. The xylem is stands there are embedded mixed into the phloem means the xylem is embedded in the phloem. So that's why there are to look like a mesh-like structure in this mixed protosteel. So this one is arranged in the Lycopodium serratum. So here these four types of the steels are present in the Lycopodium step. Depending on the species, depending on the arrangements of the xylem and phloem. Okay, clear? Okay, understood?
Yes, sir.
Okay, thank you. So this one is compulsory question in your exam point of view. Lycopodium stem anatomy. Write down with the label diagram and write the different types of the steels. Is actinosteel, plectosteel, mixed steel. These types you should write and draw the diagrams also with examples. You should write.
Okay, sir.
So and the life history of this Lycopodium here, you can see this one is the sporophyte is there. So here this one is the sporophyte plant is there. They bear the cones or stabales. So these sporophyte, there are called club masses. Already you know that one. Club masses means these bear the clubs are there. Means on the apical part of the shoot or stem, they bear these sporophylls. So these sporophylls are aggregated and there are large in size, which is to form a stabales or stabale. The sporangia consists an outer jacket cells and inner sporangium or archisporial cells are there. So see here, this one is the sporophyll. So central axis is present on the sporophylls on the adaxial surface. Okay, means lower side. They bear the sporangias. The pear-shaped sporangias are there. So that sporangia, they have the sporophylls. See here. When we take this one transfer section of the kidney shaped sporangias are there. So in the sporangia, spores are there. Okay. This one here. So eusporangiate condition is present. Okay. So in this process development sporophylls and jacket cells, there are the eusporangiate condition is there. Here, that the innermost layer of the development nutritive tissue is there. That one we call as the tapetum. And during the development of the tapetum, these cells are degenerated to secrete the uh, nutrition food. Nutrition food we produce. So these sporocysts are round and separated and floated on the viscous fluid inside the sporangium. So after these sporocysts undergoes to the meiotic division to produce the spores. Haploid spores. Four tetrad haploids are there. That is in for tetrad condition is present here. This one is the tetrad. Tetrad condition is there. Means each spore mother cells means sporocyst or spore mother cells undergoes the reduction division to produce the spores. Tetrad haploid spores are liberated or generated. When the complete maturity of these spores, the spore mother means sporangia wall will be breaks and to release the spores into the surroundings. Means stomium is breaks down and split that stomium wall and that spores are liberated. So each spore round and very small in color and around very small in size, which is the very thin wall is present. Okay. So after these spores are settled on the suitable areas, and these spores are germinated to give as a prothallus. Prothallus are nothing but the primitive gametophyte, initial stage of the gametophyte. Okay, so these spores divide to produce the two phases are there. That is the apical cells. Two cutting phases are there. That after that number of division to develop as a complete gametophyte. So here there are the two types and gametophytic stages are there. That is the Lycopodium serratum and Lycopodium antinum. Here number of steps are there. Okay. So when the coming to the reproduction system of these Lycopodium, see here. So this one is the Antheridia and Archegoniums are there. When the gametophyte development, they are to bear the reproductive organs on the gametophytic plant body. Okay. So this gametophytic plant body, the antheridias are the reproductive organ of the male, means which is covered a jacket layer and inner sperm cells are there, sperm mother cells and androcyst cells are there. So these androcyst cells again to developed as a anthrozoids, they have the biflagellate anthrozoids are there. So when they complete the maturity of these anthrozoids, there are the antheridia wall will breaks and to release it to the their surroundings. The another one is the archegonium. So the archegonium is embedded in the prothallus and to form a neck, means flask-like structure is there. Long neck is there, which is to protect the jacket layer, and the neck canal cells are there. Around neck canal cells, 4 to 5 neck canal cells are there. Phallon-based venter canal and egg cell is there. So when the maturity of the archegonium, the neck canal cells are degenerated and they pass they have the means path for the anthrozoid. So ventral canal degenerated and to form. There are to fuse with the anthrozoid to form a zygote. So after zygote to develop as a embryo. Okay, clear? So here, see here the life cycle of the lycopodium. Okay. So life cycle of the lycopodium here. There are the embryo is there. So here the embryo will develop. So after the embryo, again based on the different types of those species, there are two stages are there. They have the serratum and campanatum. Two types of the developments of embryos are there. So first of all, here Lycopodium sporophyte, they bear the strobilus, means cones, nothing but cones or strobilus. These bear the sporophylls. On the sporophylls, sporangias are there. The sporangia, they undergoes to the spore mother cells. The spore mother cells undergoes to the reduction division to produce the spore. Applied spores are produced. Okay, applied spores are produced. So the spores then to germinate to produce as a prothallus or gametophyte, which bear the antheridia and archegonium. They are to produce their respective gametes. After fertilization to form as a zygote, the zygote to develop as the embryo. After the embryo again to develop as a, a new sporophyte. Okay, clear?
Yes,
Lycopodium.
sir.
Thank you. So next coming to the Selaginella. Okay, the cones, Lycopodium cones, LS of the cone, they are asking compulsory in short question. Okay, Lycopodium, Sobalus are cone. They are asking in short question. And another type study here, Selaginella. These Selaginella coming to the Lycophyta or Lycopsida, class is Ligulopsida, order is the Selaginella, family is the Selaginellaceae, genus is the Selaginella. So these Selaginella is widely distributed with around 700 species are there. They can grow on the tropical and subtropical areas. So here some are the cultivated as ornamental plants. Hmm? Some are the cultivated as ornamental plants. This Selaginella is called club mosses or spike mosses. So here also club-like appears are there on the apical branches. They bear the stabili or core. So that's why the sporophylls are aggregated to form a stabile. So that's why we call as a club mosses or spike mosses. Means spike-like structures are appear. So that's why we call as a spike mosses. So here there are some xerophytic conditions also appear. Some are the grows on the moist area. When we take the here external feature of these Selaginella, see here, this Selaginella is the morphology based on their habit and nature of the leaves. Selaginella here two groups are there. That is the Stachyandrum and Euselaginella groups are there. So Stachygynandrum, that is the leaves. There are the difference and called as heterospilous. Stachygynandrum species, the leaves are here different are there. So that is the heterophilous condition is there. And here Euselaginella, only one type of the leaves are there. So that's why homophilous condition is present and dorsiventral symmetry is there in Tachygynandrum species, and here isosymmetry is present in the huge Selaginella. The leaves are arranged in row manner in the Tachygynandrum, but here spirally arranged in huge Selaginella. So there are the erect branch are there, very prostate condition is present in the Tachygynandrum. Erect plants are present in the here use Selaginella plant. Okay. So here the leaves are spirally arranged on the stem. The root system is well developed or advantageous root system is there, and the stem is prostrate and are creeping on the stem. Some species here erect stem is also present. Here that leaves here especially the ligule is present on the dorsal surface of the leaf. See here, a small appendages is there, that is ligule is present. Leaves are small and vary in shape and size in different species based on the species. The shape ranges from round, oval, or sometimes linear condition is present. The leaves are arranged in generally four rows in Stachygynandrum species. whereas the spirally arranged and used Selaginella species. So here there are the some membranous tongue-shaped ligule is present at the base of the leaf on the adaxial surface of the leaf. Means adaxial means lower area. Okay, the ligule is well developed to younger leaves and became conspicuous in older or mature leaves. Young leaves only they have the ligule is present. Ligule is a small scale-like appendage on the adaxial surface, means ventral surface of the leaf. Okay. Then internal structure of these Selaginella is here. When we take the here rhizome transfer section, here there are the epidermis is there, which is a outermost layer, and epidermis is covered a thick cuticle, covered a very thick cuticle is there, which is protecting xerophytic condition, and below the epidermis is the cortex region is there. That cortex is differentiated into outer, middle and inner. So outer is the sclerenchyma tissue is there. So sclerenchyma tissue is present. They give the mechanical support to the plant, and which is called as a hypodermis. Means hypodermis or sclerenchyma tissue is there. So sclerenchyma tissue is there. So that's why they give the mechanical support. The inner one also they give the mechanical support. Sclerenchyma tissue. Middle one only to give the to storage region. And here the xylem is present uh, and phloem is present around the xylem. Means xylem is surrounded by the phloem. Means here protoxylem is exarch condition is present. And leaf anatomy, when take the leaf anatomy, in the outer epidermis is there, a number of stomata and upper and lower surface of the stomata. So here mesophyll cells are there, compactly arranged, and spongy cells also, palisade cells also present, and here mesarch condition is there. And when take the here stem, which is the stem is the transverse section of the stem. Here three zones are there, that is the epidermis, cortex and stele is present. So here cortex is the means epidermis is a single layer epidermis is there, which is the outermost layer, and which is covered, the cuticle is present, thick wall cuticle is there, and below the epidermis cortex is there, that is cortex is differentiated into three zones are there, that is outer cortex, middle cortex and inner cortex. Okay. So here the outer cortex is made up of the sclerenchyma tissue, which is give the mechanical support, and inner air chambers are present. So number of air chambers are there. Each air chamber is differentiated into or divide a single layer trabeculae. Trabeculae is present, and the central is the pericycle is there, and that is the pericycle is present, means below the epidermis, below the endodermis, the pericycle is there. So pericycle, which is to cover the stele. So stele is the xylem is central time. Xylem is surrounded by the phloem, and the pith is the here some small amount of the uh, pith is not present here. Totally, the pith is absent. So that's why the stele is protostele condition is there. And the reproduction system of the here, the reproduction here. So here in the life cycle here, the sporophyte of the Selaginella, they are present in the different habits are there. So here, two types of these spores are produced. So here there are the two types of the sporophylls are there, that is megasporophylls and microsporophylls. So means that's why it we called as a heterosporous. So two types of the spores are produced in the sporophyte plant. So that's why we call as a heterosporous. So in the vegetative leaves, maybe differ some slightly is different. Okay. So here, see here, there are the two types of the spores are there in that picture. Here there is megaspores are there and microspores are there. Microsporophylls and megasporophylls are there. Both the mega and microsporangias are very short stalk is there. So megasporangia are large in shape and parallel compared to the microsporangia. And so here the two types of the spores are there. So that's why the heterospore. So here the here the female gametophyte develop at the ending of the megaspore. It is consist of the apical primary prothalial tissue on the basal archegonia. The archegonia on the extremely reduced, they have two tied neck and one neck canal cells are there. See here. See here. So after they complete the development of these one to develop as a here to develop the means megaspores and microspores. After there are to develop as a fertilization to develop as a zygote. Okay, after the zygote to divide some number of anticlinal and periclinal division to formed as a embryo. So after that embryo again to develop as a to complete sporophyte means dichotomous branching slowly dichotomous branching is developed and young sporophyte will be developed. Okay, clear? Collagenella life cycle. Okay, clear?
Yes, sir.
Thank you. So here this one is the spores are there. Megaspore. Megaspore structure is there. Okay. So in the Salaginella, they are asking here life cycle. Maybe sometimes they are asking the life cycle. Okay. So only few minutes you stay. After that, we have to leave the class. Okay, clear? All of you understood? I means xylotum, lycopodium, and Salaginella. The external means structure, internal structure, morphology structure, and life cycle. In the xylotum, synangia is compulsory asking. Synangia of the xylotum. 3 marks means short question they are asking. Short question, synangium is asking, and lycopodium cone they are asking. In essay question, lycopodium stem. Yeah. Okay, clear?
Yes, sir.
Thank you. Thank you. Only few minutes you stay. After that, we can.